This is a production of Cornell University.
Thank you, Kelly, for a long introduction. And thank you, Shipping, for hosting me today. Thank you all for being here. Today, I will talk about last 4 to 5 years of my research in blueberry. Not mine, our research in blueberries. Blueberries are one of the fewer crops that have been domesticated in North America. They are perennial shrubs. The current economic value of blueberries is around $1 billion. Blueberries have high amount of phytocompounds like anthocyanin, phenolic acid, and flavonoids, and so on. So health benefiting compounds, along with vitamins and many trace elements. Many research studies shows that regular consumption of blueberries helps reducing the wear and tear of age-related issues. Because of this increased awareness of blueberries, there is an increasing demand. Demand also is increasing for last 2 decades. So you can see here, there is a continuous demand for blueberries consumption. The green line is the consumption has been increasing from 2000 onwards. Even though production also has been increased to meet the demand, we are still importing a large portion of blueberries from other countries. So that leaves us scope for further expansion of blueberries, also crop improvement. I just wanted to give you a brief introduction here about 1 second. How can I get rid of? This one. Sorry. Can I help them?
Yeah, yeah.
Also, I can see the slide there. So here I just wanted to show you a brief introduction of different blueberry species exist, and like slightly about taxonomy. Blueberries fall under the family Ericaceae, and the next hierarchy in this one is the genus Vaccinium. Under the genus, there are more than 200 to 400 species are there. But typically, what we call them as the blueberries are from the section Cynococcus. It may be around 200 species around. The high bush, there are few terminology. I don't want to take up too much time, but it's important to give you some introduction. The cultivated blueberries are called high bush blueberries. Basically, they are larger plants, easy to cultivate, and for horticultural maintenance, easier, and bigger fruits, so on. So the low bush blueberries are from Vaccinium augustifolium. What you see in the frozen section in the grocery stores, and also what blueberries that you see in the processing foods are all from the low bush blueberries. What you see on the fresh market, the fresh blueberries that we get, those are high bush blueberries. High bush blueberries are typically grown in Canada and northern parts of the United States. So many of the cultivated blueberries are tetraploids. However, cultivated blueberries are corymbosa. They're mostly what we see in the varieties that are tetraploids. However, we also have diploids and exoploids. The low-bush blueberries mostly are heteroploids. What we call a southern species or Darovai, Virgatum, and Tenellum. They are mostly low bush and grown in southern subtropical regions of the United States. So low bush, we are considering this. I'm talking about the higher high temperature stress tolerance. So they may have some genes that can contribute to the high temperature tolerance. Also, like originally, blueberries were grown in northeastern parts of the US and Canada, but however, now we are growing them in Kerala, Carolinas and Florida and so on, so places, and also in South America, because we have developed southern high bush types also. So southern high bush types we developed by intercrossing the northern high bush with southern species. So blueberries, I was talking to shipping in the morning, how the blueberries are acid-loving plants, how you can grow. So they are very finicky plants to grow. And the best ideal temperature is in between 4 and 5 soil pH temperatures. There are also shallow root system. So since they have shallow root system and also likes moist soil, we have to have a well moist soil, but good drainage also. So if you have a stagnant water, they cannot survive. So those are few tips to grow blueberries. And they also have a long juvenile period compared to many other plants. So you can able to see fruits and flowers after 2 to 3 years of cuttings. And or if it is coming from the seeds, minimum 3 years. But you can see a good fruit yield once they attain 8 to 10 years of age. So what is the need for improvement of blueberries? We are already growing everywhere, and what is the need? But since the blueberries are relatively new crop, I say that because we domesticated only a century ago, around 1920s and that time. So from that time only we started applying our selection process. So there is a scope for a lot of improvement. And also last 20 years, we can see expansion of crop geographically as well as the consumption. Of course, if we think about any crop improvement, we first think about higher yields, right? So we want the farmers to be profitable. And then fruit improvement, fruit quality. So fruit quality here, we talk in a different languages here. So when we talk about fresh market, we wanted to have a sweeter food, the good appealing one. Also, we should be able to handle very well for transportation, so and so qualities. But if we are talking about ocean spray or other juice making companies, they would like to have more acidic and within the acid balance juice, because we don't want to add too many additives now in our juice products or any products. So they would like to see a different ratios of acid profile in the fruit. And as a consumer for fresh market, we wanted to have a sweet fruits and appealing one. And the major, and in addition to that, the major input cost for growing blueberries is harvesting, right? So for fresh market, we still, in majority of the cases, we are harvesting by hand. That is the highly, one of the costliest input that we are investing. So far, what we selected, blueberries are for looking at the yield, taste, so on and so qualities, not meant for using machines to harvest. So there is a room for improving plant architecture that can allow easy huge harvesting with machines, so which can include the inflorescence. Now, if you have a close compact inflorescence, it will not be possible. You may lose a lot of green fruits. And also, if the whole inflorescence are ripening at different stages, also you will not be getting as much yield as possible. So we would like to see some open inflorescence where, and also all the fruits can mature, at least majority of the fruits can mature or ripen at the similar time. So that would be an ideal architecture of the blueberries for mechanical harvesting. And when you have intense cropping for any crops, you'll have pests and diseases. So naturally, we have room for improvement over there. And my interest last 4-5 years is to look at the high temperature stress tolerance. And there have been a lot of studies done for freeze tolerance. I know these are two contrasting things, but there is another day of talk for freeze tolerance. But for today, I'll focus on high temperature stress. So remember one thing is that the northern high bush blueberries are originated in the temperate regions, but now we are cultivating in beyond their natural habitat. So California, many places, almost 40 states in the US, as well many states. And here, just to prove, not to prove that, in addition to that, we can see in this graph the number of heat waves per year was in an average 2 heat waves per year in 60s, but now it's almost average 6 heat waves per year that we have been looking at. And also the length of the heat wave season it was almost 20 days per year. It used to be, but in the 60s, and now it is almost 70 days, more than 2 months. These 2 months coincides with where the fruit flowering and fruit development is happening. It's very crucial for northern high bush blueberries to have some innate resistance to the high temperature tolerance. So here is what happens when, also we see even this summer we saw in the places where we never had like 45 degrees temperature in Northern Europe and many places here as well. So what happens when you have 35 and above degree for a couple of days or 3 days? The fruits, wherever the sun exposure happens, will become softened and then shrivel, and then as other the physiological maturity happens, they become like dry fruits because of necrosis. And the typical symptom that you see on the leaves are scorching. So this also has been published in one of the papers, not from our lab, but in it's a background information for this study. We were looking at whether when we thought about high temperatures, because not much research has been done in this high temperature stress area for blueberries. So we were looking whether the existing varieties exhibit any variation for this trait. So in this study, they tested on 4 different varieties and also looked at the electrolyte leakage and the MDA accumulation. So here you can see the susceptible variety Brigetta has a higher amount of electrolyte leakage when temperatures hit 40 degrees. The same way, higher levels of MDA in Brigetta, susceptible one, with higher MDA levels at 40 degrees. But until 30 degrees centigrade, you don't see a big difference, a significant difference. Same way here, for other photosynthesis and fluorescence parameters, you can see the difference among the genotypes when the temperatures reaches 40 degrees. In the summer also, we saw 43 degrees in France for 3 days or 4 days. So those are the times critical for blueberry, and also it coincides with the fruit development. So this is seedling or seedling tolerance. So sharp blue has a fewer plants we lost, but in the Brigetta, almost 43% of the plants was gone with high temperature stress. So with this background, we thought maybe we can look for candidate genes um, from the genotypes that we have, or we can make new crosses and then see alleles or genes that can help with the temperature stress tolerance. So we decided we will go with diploid species. So that way it's easier for us to do the genomic analysis. And also when we started, we didn't have a reference genome for blueberries. So we thought with the best strategy to use a diploid genome, diploid species. So Corimbosum is one of the parents, and the Doraway is one of the parents in our process. Doraway is from southern, collected in Florida, southern states, and Corimbosum is from New Jersey area, locally collected from those species. And you can also see the difference in the leaf morphology. The Corimbosum is a deciduous plant. It sheds leaves in the winter time. Doraway has a waxy coating, and the leaf orientation is different that can help withstand with high temperatures. So in the back side, you can hear me, right? In the back. Yes. One of my graduate student, previous grad student, and she looked at it. Is there any physiological anatomical differences are there between these two parents before we initiate studies? So she imposed 40 degrees, mimicking the daytime temperature until 40 degrees, and then collected leaf samples at say 0, 6 hours, and 9 hours, and then looked at the scanning electron microscopy. So what you see on the red dots, here is chlorophyll, structure of the chlorophyll, and the gray color is the stomata. Doraway, the intensity that you see, red intensity, that is chlorophyll structure. So at 40 degrees, 0 hours, 6 hours, and 9 hours, so we can assume, or I can see that the chlorophyll is almost intact. Intact, how I can say that you can see high intensity red structures here. Whereas in Corimbosum, you can see small, tiny dots everywhere. So what happened there, at least our theory is what happened, the chloroplast might have burst open and the thylakoids and stroma, they just popped up. So how I explain that is that chlorophyll is packed tightly like a small purse with stacks of coins, and you have a high pressure with the high temperature because of the cellular damage that's happening. So the thylakoids may spill open like how the coins spill open. So you don't have the tight purse anymore. So that's how you have smaller dots of tiny dots you can see on the scanning electron microscopy in the Corimbosum. Whereas in Doraway, from the southern species, you can see little almost intact chloroplast. And also we looked at the stomata, stomatal opening. In Corimbosum, you can see there's almost a similar opening as we progress with the temperature. But here in Doraway, as we go out by 9 hours or 6 hours, you can start the slightly closing up the stomata. That's the indicator of tolerance to the high temperature stress. I think these are a good candidates for studying further. And we took GWAS and gene expression studies to find genes. So within 5 years, it's impossible for me to establish crossings and study. So I collaborated with Dr. Nick Wotsa from Rutgers. He's retired now, but he was gracious enough to share his research experiments and share his germplasm and the population with me for my studies. So OPB, what you see here, the parents of OPB, different clones, 15 and 88 are different clones of OPB, is Corimboli. And then different clones of Daroway. So we made two sets of crosses. Since also I wanted to warn you here that these parents are not homozygous like what you see having rice and other soybean. So you have a homozygous parents, but these are already heterozygous. So to start with, and then F1s we cannot self them because they have a they are highly cross-pollinated species. Highly self incompatibility is there. So you have to cross them to get a seed. So we developed pseudo F2 population. We had around almost 1,000 F2 pseudo F2 population, but we have for this jivas we utilized around 260 to 300 plants. So here are the F2 that we set up the experiment with 40 degrees on Krishna did come. He is the main person working on this project, another postdoc in the lab. So here itself, you can see in the visually, it's not the shade. So there are some plants with good tolerance, and there are, you can see this spotting symptoms in between the F2. And here is the one with Dr. Otsa and his group with us, with him, we do fruit acids and phenotyping fruit acids and sugars. So for GWAS phenotyping, we took both sides. One way, what are the first responses will happen in the plant, plant damage when exposed to the temperature. The other way, and other side, we also measured few things, few parameters that inherently what defense mechanisms plants can exhibit. So as a biomarkers. So Krishna, Dr. Krishna, he's from our lab. He's measuring different chlorophyll and the fluorescence parameters with polypin and chlorophyll for phenotyping. So what we see after 4 days of exposure to 40 degrees, what you see here, some of the plants, tolerant plants that you see on the top row, you don't see any scorching. However, in the bottom row, you can see the scorching effect. Sorry, these are the sensitive plants, and the top row can be resistant plants. The F2. And then once we have collected data, we looked at the frequency distribution, how they look in the population. So you can see normal distribution for majority of the parameters that we took. And for the quantum yield, we have the frequency distribution. Also looked at the box plots to see the range for this population. It's pretty decent for to go with the jivas. And again, box plots for other parameters. We also looked at the heritability because this is our first time we are working with this crop and these parameters, whether they are heritable, if we select for any of those traits. Since we have a decent heritability, so we still hope for, we're going to have a hope for better improvement of these traits. And this is regular protocol with the Illumina sequencing and so on. We are with similar protocol. We also have a paper published for a different project. So from this sequencing for this population up to 60, we had around 1 billion reads, raw reads, and then mapped those read treats with Draper tetraploid genome. So we were able to get the reference genome by that time. We had 85%, almost 85% of mapping. And after filtration, you have 126,000 SNPs identified from this sequencing. When we look at the Manhattan plots for each of the traits that we have seen, there are around 1,323 SNPs that are associated with these traits that we measured. And from here, what we did, we selected SNPs that have that are associated with more than one trait to go forward to make it easier. So with that, we have around 245 SNPs that have been associated with more than one trait and that we measured. And out of this, 172 of them in the genic regions of the genome. Almost 40 genes are in the chromosome 1, and 11 and 12 also have a considerable genes that are associated with multiple traits or multiple parameters for the heat tolerance. So these are some of the candidate genes from those what we selected from the GWAS. At the same time, simultaneously, we had another student, grad student, who was working on gene expression studies. So we used the same parents from Corimbosum and Delaware for gene expression studies. So she treated plants with 45 degrees at 9 hours, 6 hours and 9 hours, collected samples. So we took this name. She also did preliminary experiment with cultivars, the cultivars that we have using northern high bush, southern high bush, and rabbit eye, different genotypes. So based on the preliminary study, we chose these time points for this project. So for a gene expression analysis. So these are typical sequencing protocols that she followed. And after assembly and filtration with the quality checks, we have around 99,000 assembled genes in a corimbosome and almost similar amount, 109,000 genes in Delaware to move forward. So these are in the Venn diagram of the upregulated genes and then downregulated genes. Then we also looked at the common genes across the genotypes and across the treatments. So what we were interested to pick candidate genes were the common genes. And we took these 325 common genes and cross-compared with the GWAS genes. Is there any, because if typically they are responding, we should be able to see here also, right? From what we found from GWAS that are associated. So we have around 30 genes that are common in both the studies. So that is a good sign. So this can be a strong candidate genes for either marker development or for further studies as well. So from this two studies, we were able to identify 245 candidate genes, and also both susceptible and tolerant F2 plants. So since these are diploids, we can transfer the traits that we identified from here using crosses with the tetraploid blueberries. So that project is underway now. We're also developing PCR markers, because every time you cannot use the SNP markers to, at least with the facilities that we have. So we are converting them into PCR markers and making them into robust, everyday usable markers to help aid in the selection process. So that's the one. Another project also I wanted to share it quickly because it just published yesterday. It's not for promotion or anything, but I just wanted to share it with you. I was talking about the second, in my second or third slide, there are 200 species of blueberries. So we wanted to see, we are, the reason also there is another study from, I don't remember, there is a Florida group or Carolina's group. Even though we have several varieties of blueberries releasing now, so for to grow them in the south or in the tropical temperate region, but most of them, the base genetic material is coming from the only fewer plants, fewer beans. Like most of the southern species have come, like Florida 4 or few other plants or few other cultivars. So exploring more wild species, there are plenty of wild species, and we have availability in different from different groups also. Exploring them, characterizing them will be beneficial for further breeding improvement programs. So with that goal, we looked. One of my master students project is now who is doing PhD in Florida. He identified SNPs with the genotyping by sequencing method, and then compared the genetic relations. And I'm really interested in this project. We found really good stuff. So just to give you a brief idea here, one of the boreal, Vaccinium boreal, collected in Canada, Nova Scotia regions, which have Rocky Mountains, alpine region habitat. And the corimbosum is collected from New Jersey, where you have swampy areas and mostly grow in that area around the rivers and creeks. And then 10LM is from Virginia and North Carolina regions. And Daroway and Mercenitis, they were collected in Florida and Alabama region. So different places were collected by Dr. Nick Watch and his group in 80s or some time ago. So I started using those genotypes because they were just growing them, and I wanted to explore more with those. You can see they can be beneficial. So in total, we used 195 accessions from each species. So I think somewhere you have those numbers also. So from those, after a quality control and everything, you have 60,000 SNPs that can be utilized for further analysis. So when we do the PCA, you have a clear-cut separation of southern types. All these are southern types, and these two are northern type blueberries. But within the northern type, boreal is here, is separated out from corimbosum. But whereas in the southern types, all of them, at least they didn't separate as distinctly as the northern type. We have my time. 13. Okay, so this is MJ tree that's based on the distance. So you will see same way of separation that you can see in the previous slide with the PCA. Again, what we think so far is these two accessions, what you have here from boreal and corimbosum. So there can be a bridge accessions. So that's we haven't proved yet, but those can serve as a bridge accessions between these two species. And this is to talk about nucleotide diversity. So we have plenty more number of accessions in Delaware. Even with that, but there is less nucleotide diversity in Delaware compared to the 10LM and boreal. So when you look at the admixture, the first one to deviate from all the rest of the group, whatever we have studying, is the boreal. So boreal here when you have it made into two clusters, you have northern type and then southern type. And then you further divide, the boreal got separated out immediately. All the southern types stayed as it is. And then after, when you make four clusters, then you have 10 LM separated out. And then after you make 5 or 6, we couldn't see the separation between Mercenites and Darovii. So we are thinking they are admixture because of their habitat, because they grow under similar conditions. There may be cross-pollination happen, outcrossing between the two species, and it's hard to separate out. And this is also very interesting in the Manhattan plots. When you see the Manhattan plots, comparing with here, I'm particularly interested here with boreal, comparing with all other species, you have a negative selection here, where you have a positive selection here with pairwise comparisons. And FST also is higher in Mercenites and boreal compared to Mercenites and Darovii. That's what you see in this graph. So the admixture is high, so you also can see FST is low between those two species. So we also looked at the migration events between the species that we studied. So boreal just stood out immediately, and then as a big event happened, the corymbosom might have formed from the originated from the boreal, and then you have smaller events from corymbosom that to other species. So it may tell us that boreal may be the progenitor for northern type blueberries, or blueberries that we call them. And we also looked for the selective sweeps. Are there any selective sweeps among the species? So in chromosome 12, we can see a selective sweep. So we choose corymbosom as a comparison because that is the cultivated one. The rest of them are wild species. So you can see a clear selective sweep here. Probably whatever genes are there in the chromosome 12 in this region are making the natural selection or artificial selection over other species. So that sweep region is around 715 kilobase pairs, and we have 32 genes that may be interesting to look for further deeply. Some of the genes that I found among 32 were some meristem maintenance, DNA repair, sucrose synthase genes. So there may be for interest for further studies and also. This is to summarize what we have. So Darovii and Mercenites, that's why, because boreal is in the Canada with Rocky Mountains and so on. So all higher altitude habitat. The corymbosom is in swamp areas in New Jersey. So there may be there is a not much possibility for cross-contamination, cross-pollination or crossing between these species, but sometimes have originated from the boreal. But whereas in the Darovii and Mercenites, because they're cohabitat between the similar places, so there may be a lot of admixture happen in those species. So that's our prediction from this study. And my collaborators, Nick Wurz, I have been talking about, and he provided the material and the knowledge about blueberries because I am very naive when I started this project, and he helped me understand about blueberries and sharing his knowledge and wisdom. and Dr. Umesh Reddy from West Virginia State, and he's my bioinformatics guru. So if I need any analysis, so he's the one I go with him, and he helps me a lot. And some of the students are not there any now. The paper that published is from Byron. He's in Florida now, and all of them are master students, graduated, and everybody is doing good. And, she's also a model now. And Hemant Arvind is here. And Tommy joined later after we took the picture, so I had to have her separately here. And this is my lab website. If any of you are interested to explore, we can stop here, and then I'm open for questions.
At the beginning of your talk, you showed us some photographs of fruit in response to heat stress. But I noticed that most of your phenotyping of heat stress was on leaf properties.
Yes.
So I'm wondering, have you found there's a good association between the effects on leaves and the effects on things like fruit set, flower set, and yield?
Flowers, I haven't started yet. That's the future study. Yeah, no, we haven't started that. So we started with, because for me, it's easier, since we have F2, they may flower different times. Concluding the experiment will become harder. So we initiated with the leaves, that way easier. We can measure all at one time, and one time exposure of the treatments. So in the future.
For the heat stress experiment you did, what was the humidity like? Was it very dry or was it very?
I don't remember, but it was 70%. It was in the growth room that we import the temperature, so we can regulate.
Is Mercenites possibly an allopolyploid? And if so, does it perhaps have the genome of corymbosom as part of it? And that would explain the admixture that you see.
To the Darwin.
Darwin.
Yeah. Allopolyploid, I'm not sure allopolyploid or not. I haven't looked at that angle. So only we thought maybe because Mercenitis has a tetraploid version also. Tetraploid version. So because of the outcrossing nature, there may be admixture happen. So don't know. Darwin also has tetraploid versions. Tetraploid version. That is the one mostly people utilize for developing southern blueberries. Florida B phenotype. So many. That's the one reason also I got interested in this project, looking at the different species, because whatever the existing, many of them, I'm not taking blanketing all of them, but many of the blueberry projects that we have, the base genetic material is the same, coming from similar or fewer sources. So maybe if we shine light on different species, we can use them for some plants.
For the tetraploid steroid suspension, does it also have the small weeds typical?
Tetraploid one, so the shape is same, but little bit bigger than. Yeah, plant also smaller than corymbosom. Plant can go up to my height, the tetraploid one, but the diploids are tiny plants. The shape is almost same.
Yeah, that's I have two questions. Yeah, the first one, you mentioned there's one gene that involved in meristem maintenance. I wonder, the question is like, it's specific to you or to shoes?
This is from the annotation. So we have to find out more.
Okay, yeah.
Not that we experimentally proved. We looked at the sequences with annotation.
But the gene that's involved in the maintenance of
maintenance.
activity.
Yes.
Yeah, so this one. And the second one is like very interesting. The blueberry. So we discussed a little bit this morning. Require acidic soil or something. My question is, what's the role of low pH or aluminum Al3+? How, I mean, this contribute to or or maintenance the growth of blueberry or promote the secondary metabolites of flavor or formation?
I haven't looked at aluminum point of effect of aluminum point of view, but I had some bad experience when we have soil. So when I started this project, we just used the greenhouse water, whatever water is available in the greenhouse. Plants are terrible; they look terrible, and they are not growing up. And then because when you just start your faculty position, you haven't rushed, right? You wanted to, and I have undergrad students working with me. And then I spent more time then we went to the test the soil pH, what the water in the greenhouse, it was like 5 something, 5.2 or 5.1. My plants are not, they're sick almost. So then we literally took some HCl, HCl diluted, and then we mixed with water, and then apply it to the blueberry pods. After 2-3 weeks, you can see they are coming back again.
So that's
So,
pH is
pH, yeah. So that I can tell with confidence. But with aluminum, we haven't tried any experiment with that.
interesting.
Definitely, that we can talk for further.
Yeah, sure.
I can send samples and we can also.
Yeah.
Did you remember, Professor, that we did one trick essay with a different pH? Even if it is pH more than 6, the plants are showing all necrotic symptoms. Even if it is lesser than 3, they are showing, even though they love acidic, but not lower than 3.
Yeah, I think one summer project for
Yeah, we did.
This has been a production of Cornell University, on the web at cornell.edu.